some software for data. So we just wanted to start with Okay. Yeah. Okay, so I thought I'd start with a little bit about my background. So originally from Australia, I studied there and did my degree in biochemistry. And during my PhD, I worked on genomics. Um, and it was sort of during my PhD, through analyzing data, I was kind of learning programming. That was my first exposure to programming. And actually, part of my PhD was developing this Python package to detect different sorts of DNA variants from DNA sequencing data. So that was some initial exposure to programming and learned a little bit through that. And then in 2018, I finished my PhD and moved to New York City for a postdoc. And there I joined a very like computational biology heavy lab that also did a lot of software development in R. And so I stayed there for about 5 years, developing some different tools for computational biology. And then about 2 years ago, I moved here to Singapore and started my lab at GIS in A-star. Yeah, so I want to kind of start this talk with some background about genomics because I think maybe many of you don't really know so much about genomics yet, since we're in the School of Computing. So this molecule is DNA. It's a very long polymer that all of us have in our cells, right? And it's composed of these different what we call nucleotides or bases. So there are four of these different bases, so A, T, C, and G, and they pair together in a particular way. So A pairs with T, and C pairs with G. And so they're arranged into this helix where you have all the base pairs sort of stacked up. And the point of this molecule is really the most important thing in your cell because it holds all the information that you have, that an organism has, right? So this ordering of bases in the DNA strand encodes information about what that cell should do. And so the really important thing about DNA is that because of this base pairing, you can actually copy the DNA strands and make perfect copies. So you pull the strands apart, and then you can fill in the other strand, the missing strand, based on the information that's in one strand because of this base pairing. So that's the sort of mechanism the cell uses when it divides, and it can copy its DNA, and then each daughter cell gets a copy, and so that information is maintained. So that's DNA. When we talk about genomics, this just means that the genome is all of the DNA that an organism has. So for humans, us humans, we have around 6 billion base pairs of DNA. That's arranged into 23 pairs of chromosomes. So half from your mother, half from your father. And yeah, so genomics is like just studying this genome, which we can kind of think of as like the software for the cell, because it contains all the instructions that the cell sort of executes in different ways. Yeah, so one question that comes up is, okay, we have one genome, right? Us humans, we have one genome. All of the cells in our body have that same genome. So how is it that we have like different cell types that are very different in their function, but they have the same set of instructions? So the way that this works is there's a sort of another layer of information that's added, which we call epigenomics. So essentially what this means is that this long strand of DNA isn't just a long strand of DNA in the cell, it's actually wrapped around different proteins and has these like complicated three-dimensional structures, which do vary across your different cells. So you have these piece of DNA wrapped around different proteins. These proteins can have modifications put on them, and the DNA itself can have modifications put on them. And these modifications can also be heritable across cell division. And so this can essentially instruct the cells to have certain areas of the DNA turned on and off in different cells, and this information can be maintained. So if we want to study the genome and understand how this genome is being used in different cells, sort of being able to measure these epigenomic marks becomes very important. Another layer that we have is kind of the output of the DNA. So the DNA shown on the top here provides these instructions, and then the way that this is sort of utilized by the cell is by first producing another molecule called RNA from this DNA sequence. So there's another piece of nucleic acid that gets produced, and then that can be translated into another thing called a protein, which is what most of the functions in the cell are carried out by these proteins, because they form these different three-dimensional structures that can have different like chemical properties. And so being able to measure the RNA in the cell is also very important, because it can give you this sort of window into like what genes are active versus not active in the cell. Yeah, so the main technology that we have in genomics is DNA sequencing, right? So DNA sequencing lets us look at these different molecules, DNA, RNA, and try to figure out what they're doing. So there are lots of different ways to sequence DNA. The most popular approach is kind of illustrated here. So the way this works is we have our DNA sequences, and we can actually attach these to like a glass slide and do some chemical reactions so that we have many copies of exactly the same molecule being produced in one cluster here. So they're all like grouped together, and they're identical DNA sequences, so sort of like a clone of DNA sequences. And then we can try to sequence these by adding these special modified DNA nucleotides that have a fluorescent group on them. So each different base will have a different fluorescence. We can add these like one at a time. So in the first round, maybe T gets added, we read out a red fluorescence. And because we have this like group of all identical sequences grouped together on a slide, that boosts the fluorescent signal so you can actually detect it in a microscope. So by going through many of these cycles, you can kind of build up this like sequence of fluorescent signal and read the DNA. And the strength of this is that we don't just do it for like one piece of DNA, but you can do it for billions of DNA sequences at a time. So you get billions of short DNA reads from one of these runs. And so what this plot over here shows is how the cost of doing this has really fallen over the past like 20 years. So around 2001, the Human Genome Project announced the first draft sequence at a cost of around $100 million for one human genome. And the orange line shows Moore's law. So for the first maybe 6 or so years after that, the cost sort of followed Moore's law. But then around like 2007, something changed, and we see this massive drop in sequencing cost. And what happened was actually this method was commercialized by a company called Illumina, and you see the cost just plummet for DNA sequencing, right? And then around 2015, we reached $1,000 per human genome, and now 2025, the cost is around $100 to sequence one human genome. So, sort of a million-fold decrease in cost over the past 20, 25 years. And this has really sort of enabled the whole field of genomics to really grow massively, because now we can do sort of population scale studies. Like in Singapore, we have this SG100K program where we're sequencing 100,000 Singaporeans, and the next stage will be 500,000 Singaporean whole genomes, and other things as well. So now we can use DNA sequencing as a way to measure lots of different things happening in the cell, like the epigenome, the transcriptome. And the next sort of biggest development more recently has been this sort of shift to looking at single cell resolution. This is what my lab works on. So in the past, the way people did things, if they wanted to study tissue, so we have like a pancreas or something, you can just take a chunk of that tissue, grind it up in a tube, and then look at that. So you can measure like RNA from that tissue, and that would just be sort of blending together all the different cell types that were in that tissue, right? So then, if one cell expressed the gene very highly, so it has a lot of the RNA for that gene, and then another cell has none of that, you just get this sort of average of the two, which may not represent any of the cells that were actually in there. But more recently, we have these methods called single cell methods where we actually can dissociate the cells and do different steps so that they're captured individually and tagged uniquely. And then we can actually measure each cell as a different observation from one experiment using different methods like microfluidic devices or other things. So the difference is that, we do this bulk assay and we get, essentially one vector. So one experiment would give us a vector where each entry in the vector would be some expression level of a gene, for example. Whereas for the single cell, now we don't get a vector, we get a whole matrix. So each column would be a gene, and then each row would be a different cell that we captured in the experiment. So we have this real like explosion of information from moving from bulk to single cell. And so, at the very beginning of the single cell field, it's still fairly early days. So around 2015, this is like before I joined the group in New York that was working on this. If you look at the number of studies over time and the number of cells reported, it's still fairly small, like not that many dots. So each dot here is a single cell study, and the number of cells is like not that much. So still fairly early days for the field. And there was some software developed. This is from the lab that I joined in 2018. But it was very sort of basic software where it couldn't really do a lot of things. It wasn't very user friendly. You'd install it from like a supplementary file from a paper. But it kind of fit the need of the time, which was that there were barely anyone doing analysis of this stuff at the time, right? But then, fast forward a few years, and then the experimental methods become commercialized. A lot more labs are doing it, and then if you look at the number of studies, that kind of starts to increase a lot. So we have a lot more studies. The scale of them is also increasing, and then there are also interesting methods being developed where people develop experimental approaches to measure not just RNA or one particular thing, but you can measure pairs of observations in each cell, like RNA and protein expression or RNA and DNA. So you have this introduction of what we call multimodal data as well. So now we don't have just one matrix, but we have potentially several matrices that are being generated. So this is like around the time I was starting my postdoc and worked on some methods to address some of these challenges. So first, this challenge that's introduced by having now many different studies. So once you have many different studies, of course you want to combine that information and compare them. So as an example, here are 8 different single cell data sets from the pancreas. So here they're all projected into a 2-dimensional space. So each point is a cell, and their closeness to each other is determined by how similar they are based on their transcriptomic measurements that we made. So you see these clumps of cells that are grouped together. Those are basically the same cell type. All right. So let's say we wanted to study the pancreas. We would want to take all this data and combine it. Right. But if we do that, then we find that there's 2 sources of variation. One is the biological variation that we're interested in studying, which is what are the different cell types? How are they different? There's this other source of variation, which is more technical variation about what method was used to assay the cells and what lab did it and all these different things. So now you see the cells grouped together. Here they're colored by their data set. And so you see lots of groups of cells that are the same color, which means they're all from the same data set. So they're grouping together by their cell type, which is what we want. but also by this other information which we're not really interested in. So what we did, we developed some new computational methods that enable you to basically identify the shared sources of variation across these different data sets and sort of remove the things that are the source of variation that are not shared, which are more likely to be the technical variation. So, we developed this new computational approach that enables you to put all these data sets together and then automatically identify what are the sort of equivalent cell states across them to enable this sort of multi-data set comparison. So then you can go from like this situation to the one on your right, where you have cells grouped by the cell type, regardless of what data set they came from, and then you can kind of start to explore from there, like what's different across these different cell types, and so on. So the other challenge that we had was this introduction of what we call multimodal data, where instead of just having one matrix, we now have two or maybe several matrices, where each one sort of measures different sorts of data from the same cell. So we wanted to address this through some updates to the software, right? So we have this software package that I mentioned before that is called Seurat. So what we did was actually rewrote this thing from scratch, basically, to really enable this multimodal analysis. So the original version was built with the assumption that you just have one matrix and that's all. So we kind of wanted to make this more flexible, so we redesigned it using these S4 object classes in R. And so the way that this works is we define this class that we call the Seurat class, which has these different slots for different sources of information. So in the class definition, you define like what type these all can be. And one of the slots is something that we call assays. And so then we have another class that we call the assay class, which contains the count matrix information and all of this. And so the change is that now this becomes a list of assays, and in our object, we can store as many of these different assays as we want. And so it uses this sort of object-oriented programming approach where we first create the object and call different functions on it, and the state of that object is updated. Different things are stored in it, and you can generate plots and do all this sort of stuff. So sort of rethinking how the software is designed to keep up with how the experiments are progressing and what types of data that you can generate. Another thing that we focused on a lot was we have to keep in mind that actually a lot of the users for the software are not computer scientists or programmers. They are mostly biologists who may not have a lot of programming background. So we spent a lot of time in developing the documentation, both like just the normal function documentation and also these sort of guided analyses that we call the vignettes. So each one of these, like we have lots of these, they sort of walk you through step by step, like different sorts of analyses that you can do with the software and how that may like work with other software packages that you have in R. And then users can just like walk through that and learn how to use it, and then even just take their own data and sort of walk through the same steps and learn how to use the package like that. So I think that was really important in getting the software like actually being used by people. And then the other thing that we did to help adoption as well of the software is we spent quite a lot of time in user support. So through like GitHub issues, and a lot of these that we get are not the typical issues that you might have of like reporting a bug, but a lot of them are just questions about like, I have this sort of data, like how should I analyze it? And so we also try to address those questions, and we'll have, back when I was in this group, we had these weekly meetings with all the developers, and we would sit down as a group and discuss a lot of these questions. Like people ask questions about how to analyze certain data. We'll kind of, some of them are very difficult questions to answer. They have like complicated experimental designs. And, we'll discuss them and think about how to respond to them. And also discuss bugs that came up that may be like complicated and think about how we need to fix them. So that was very, very valuable to have as well. So kind of looking forward, this plot I showed earlier, if you extend to 2025, you see that the scale of single cell experiments just continues to grow. And this is kind of correlated with the falling sequencing cost, right? Because if sequencing cost halves, then you can just sequence twice as many cells with the same budget. So now we have a lot of data sets above 100,000 cells and now even up to 100 million cells. And so this creates a whole new set of issues, particularly things like in R, the sparse matrix class that we have is encoded with 32-bit integer. So you can't have more than 2 to the 32 non-zero elements. So at a certain point, you just can't create a matrix that large in R. But even that aside, there's a huge RAM requirement for that size matrix. So do you have a question?
Why not back against the TGF file?
Sorry?
Why not back the data in the TGF file?
Yeah, exactly. So a lot of what we're doing is working on how to do disk-backed computation. So HDF5 or there's other methods like this package BP cells is more customized for like sparse matrix where they have this bit-packed matrix format. And yeah, the idea is that most of the data lives on disk and then you read in small chunks at a time and operate on that. And that's much more scalable. Yeah. So, obviously you can have like 64-bit sparse matrices as well, but then you're still going to have this problem of needing a ton of RAM. So we're trying to build these methods of using like disk-backed computation, even doing things like selective sampling of these cells, because you may have, a tissue that you're looking at where 90% of the cells are one certain cell type. And after a certain point, you've sampled that enough, you can kind of throw some of those cells away, but you want to keep the more rare cells. So you can do sort of selective downsampling and reduce the size of your matrix a little bit that way as well. But for me, especially after leaving the lab in New York, my lab here is focused a lot more on the epigenomics and single cell epigenomics. So this is a field that is maybe a few years behind like the single cell RNA field, just because of the technologies were developed a little bit later. But so this basically measures different types of epigenomic marks. There are lots of these different things, but kind of gives you some readout of different DNA regulatory elements, how they're active and different things like that. But the data comes with a whole host of other challenges because now instead of measuring, say, 20,000 genes, which you would have from a transcriptomic experiment, your data just spans the entire human genome, which, like I said before, is 3 billion base pairs, right? So the scale of the number of features that you have is much, much larger. You can kind of subset that to around 200,000 regions of interest, but it's still like 10 times more than you have for RNA. And the data is even more sparse because now you're measuring DNA, which we know we have 2 copies of the genome in each cell for humans. And so most of the measurements that you have are going to be like 0 or 1 and extremely sparse. So like more than 90% of the entries in your matrix is going to be 0. So there's different challenges associated with that. But in my lab, we've developed this more specialized R package specifically for epigenomic analysis, and we kind of built on this Seurat package that I used to work on through this class inheritance framework that R has. So in the S4 classes in R, you can define a class that inherits from a different class. So this means that it has everything that class has, plus some additional things that you can define. And then if you have methods that are defined for that parental class, all those methods will work on your new class. But you can add new methods that will work only on your class as well, that are like more specialized. So that's what we did here. So we have everything that the standard assay has, like you can see in this plot in the middle, like everything the normal assay has, plus some extra stuff that's like more specialized for epigenomics. Um, and so the advantage of this is that we don't have to rebuild like all this stuff that was like in common with the standard assay. And also, the Seurat object is sort of a collection of these assays. And so for multimodal data where we have like epigenomic measurements plus something else, we can very easily put these together in the same object. And then this helps us build some computational methods that uses information from all these different assays. So if we have epigenomic measurements and RNA measurements, we can think about some computational methods that would use all that information. And it's easy to implement in the software because it's all in one object. Okay, and so yeah, this is just some example of like what you can do with a package. These are the two-dimensional representation that I showed before, right? Each point is a cell. We can also have this other sort of representation now where we're looking at a certain area of the genome, and you can see in all the different groups of cells where there's sort of like a pileup of the reads that we sequenced from those cells. And this would indicate that there are certain regions of the genome that are open and being used in certain cell types, but not others. And we can study what that means. Okay, and then so more recently, something that we've been doing is a little bit moving away from R and using other languages like Rust for more speed and memory efficiency and other advantages as well. So we built a couple of tools in Rust. These are for more like pre-processing steps. I think R is still good for like interactive analysis and statistics and this sort of stuff. But we have some functions to say, take your raw data and generate that count matrix that you start your analysis with. And so in this plot, the blue line is the old R implementation that we had. And then the brown lines at the bottom are the Rust version. And yeah, so you can see that the scalability is much better with Rust. And it's not only the language, but the way that this method is implemented is different and is just thought about a little bit differently. I think the scalability, the memory management in Rust is an advantage, but also it's very nice to have this package that you can compile down to just one binary that someone can just download from GitHub and then run it on their machine and don't have to install anything. So all the package management and everything in R is actually very complicated. It takes a long time, and it's a big advantage for people to just be able to run your tool without any like Rust doesn't even need a runtime. It just works. So that's nice. And then also for like long-term sustainability of the software, once you have this Rust code that works, it kind of works in perpetuity. Whereas for R, that's not true. So like our R package, we have to keep it updated because the dependencies will be updated, and that can introduce like some errors in our package, which we then have a certain amount of time that we have to fix it. Otherwise, our package will be removed from the R package repository. So even if we don't add any new features, there's always a sort of some work that has to be done to maintain this thing from just not breaking.